Okay, so to continue our talk on natural selection and evolution, we're going to go and revisit Hardy-Weinberg just for a little bit, not long. I'm not going to show you guys how to do every single problem. We might do one or two just as an example and then move on. So in Hardy-Weinberg, if you remember this, we had our P's and we had our Q's, and the P was our dominant allele and the Q was our recessive allele. And what we were able to find out was not only just the frequency of our P's and our Q's like we did last time with using the genotypes of a population, but we were also able to see, okay, if I have P, if I have Q, what percent of the population is homozygous dominant? What percent is heterozygous? What percent is homozygous recessive? So the thing about Hardy-Weinberg equilibrium, when we're using those two equations, and if you remember, our two equations are P plus Q equals 1. In other words, if we have them as decimals, add them up, and it would equal 1. If we have them as percents, add them up, it would equal 100%. And our other equation was P squared plus 2PQ plus Q squared equals 1. And we use this one because we were able to do a Punnett square for two heterozygous individuals where P and P here would be P squared. This right here would be PQ, PQ, and Q squared. That's how we got this equation, because we got our two PQs right here for our heterozygous individuals. So it's going based off of crossing two heterozygous. So when we have these two equations, we are able to use them for our population to determine whatever we want to find out, whether it's alleles, whether it's genotypes. We have certain criteria for Hardy-Weinberg equilibrium in order for those two equations to be used. And this goes with the second bullet point here. If a population does not meet the criteria of the Hardy-Weinberg equilibrium, it can be concluded that the population is evolving. So, with these two equations, guys, it's talking about any single solitary moment for our population. The next moment might have changed those two equations because we might have had a birth. We might have had a death, and in those, we actually changed the whole population. We changed the gene pool. It might be adding some, it might be leaving some. So we have to assume for Hardy-Weinberg equilibrium that evolution is not happening. I like to always think of it as, think about it at any instantaneous second of that population. In the next second, if a birth happens or a death happens, our Hardy-Weinberg number is actually changed. All right, so I kind of went over this. Sorry, I was a little long-winded on the first slide there. So we're going with the assumption that our population is remaining constant. Like I said, at one moment, we might have a number. At the next moment, we might have a different number. For Hardy-Weinberg, we're assuming everything stays the same from generation to generation. Okay, so I'm going to skip these ones, guys, because this was the example we went over last time with the P's and the Q's. All right. We can use those P's and Q's, put them into P squared, 2PQ, and Q squared in order to determine what our genotype frequency is. So our P squared number, these are our homozygous dominant individuals. Our 2PQ, and when you're doing this, you do got to put the 2 in there, so we got to do 2 times our two numbers. These are our heterozygous. And then lastly, our Q squared, this is going to be our homozygous recessive individuals. So we could tear this equation apart into 3 different pieces and actually just use one piece of it to determine the frequency of our, here's P squared, so that's our homozygous dominant. Our 2PQ, there's our heterozygous, and then our Q squared for our homozygous recessive. And I know we already went over that in the past, guys. Okay, so here's the long way of it. We can skip all of this just because I already went over that, and you guys, I think, are pretty good on Hardy-Weinberg because we did it in chapter 12, right? All right. So again, for Hardy-Weinberg, guys, we're assuming that the population is not evolving. Is this good for real populations? No, it's not, because we change over time. The second someone dies, their genes leave the gene pool. The second someone is born, their new genes enter the gene pool, and those genes might not be the same as someone who just died. So here's our 5 conditions that must be met in order for Hardy-Weinberg to stand true. In other words, for the population to not evolve. So we can't have any mutations in there. So that's our first one. Second one, we have to have random mating. And this one always confuses students, so let me explain to you what random mating is. Random mating is not something we usually do. We actually do non-random mating. It's like, oh, hey, look, that, I don't know, we'll just say tortoise. That tortoise right there has really beneficial genes. If that tortoise has very beneficial genes, that tortoise is going to survive. It's going to reproduce more and more. That's non-random mating. Random mating is like, hey, look at that turtle with the 3 legs. It has the same chance of mating as the turtle with 4 legs. Okay, that's random mating. Obviously, the turtle with the 3 legs would not mate as often as the one with the 4 legs. So the third one, no natural selection can occur. Fifth one, we need an extremely large population size. This is so that when we are going through all this reproducing, going through all this mating, we are not changing up the numbers drastically. And then lastly, we can't have gene flow. In other words, we can't have people coming into the population or leaving the population, because if we do, then that's going to change our gene pool. So in other words, if we go back to the example I had from Africa and Ireland from last time, we cannot have an Irish person with red hair going to Africa, because that would completely change their gene pool. All right. Okay, we're good here. So when we're talking about Hardy-Weinberg equilibrium, there is one amino acid called PKU. You can see here. So some babies are born, and it's not very prevalent, but they're born with an overexpression of this amino acid. It's not a big deal. Their life expectancy is not shortened or anything like that. But again, it's not a very big prevalence with babies born. We can assume that this is really, really close to Hardy-Weinberg equilibrium because it doesn't mutate often. It doesn't increase or decrease mating, which is kind of nice. Natural selection can only act on rare homozygous individuals, and as long as they eat right, they're good. We can use a large population to test for this. All right, there won't be too many in the population that have it, and migration has no effect because it's every part of the world has a little bit of the PKU disease. So we could set this up for Hardy-Weinberg equilibrium just in normal everyday life. And you guys can see here, Q squared is 0.0001 for PKU. So it's 1 per 10,000 births. That was since this book has been published, actually. It actually went down since then. They estimate it to be roughly 1 in every 20,000 births now. So that number is even lower. But given that, you guys can see that our Q value, we got our Q squared here, which is our homozygous recessive individuals. We would have to take the square root of that to get the Q, but the Q value is super, super, super small. So it is only, 1% of all of these alleles are recessive. So we have a very big population as homozygous dominant and a very high population that is heterozygous. Very, very small, which is q squared. All right, so let's get into genetic drift and gene flow. Here is an example of something called DDT. So what was going on? Back in the day, we had mosquitoes, and they were bringing about malaria and different things like that. But they got this DDT, which it was an insecticide, and they used it in agriculture. Now, what happened was the allele that confers resistance to DDT actually increased in the population of insects. So the DDT was actually ineffective to kill these bugs, which, yeah, that's bad for us at least. For the bugs, that was good. All right. So with that example, guys, we had the alleles actually changing based on what was going on with the population. Obviously, DDT was killing the insects. So the insects that had the allele for DDT resistance, they increased. The ones that did not, yeah, they died. All right. This is an example of genetic drift. So we can have some type of occurrence that happens, and it completely changes the population. All right. So DDT came about, and it killed a lot of the mosquitoes. All right. But genetic drift can also be by chance. An example of this is people stepping on bugs. All right. Obviously, we are more powerful than bugs, no matter what. We're more powerful than them. So if we were to, you see beetles walking along the road, you just step on a beetle. That beetle that you stepped on might have been the most fit individual in that beetle population. Well, you just took out the beetle, so then you took out these dominant favorable alleles that went along with the beetle. So yeah, that's bad because you just actually took evolution backward a little bit. You actually were not following natural selection, because natural selection says that the most fit individuals survive and reproduce, and the ones that are not fit, they die and they do not reproduce. All right, genetic drift doesn't work like that. So with the DDT example, a lot of the bugs died with DDT. Maybe the ones that were the most fit didn't have this DDT resistant gene, so it wiped out a lot of those favorable traits because it changed the population. Now, the most favorable trait was this DDT resistance. All right, so all the other traits, it really didn't matter anymore. Okay, genetic drift is really affected in small populations. So once we get rid of the genes in small populations, it'll change the gene pool very, very, very quickly. All right, next one, founder effect. This occurs when a few individuals become isolated from a large population. So we have a population. That population then breaks off, and we have a smaller population, and that population will reproduce and make pretty much a new colony, I guess we can say. So when does this occur? Let's pretend we have a new island. It arose or broke off, one of the continents. We have this new island. It arises, and then evolution starts to occur on that island. That's the founder effect. What can happen over time based on the founder effect, excuse me, is we can actually have the two populations. They might be the same exact species at the beginning, but they might drift further apart from one another with respect to their traits and adaptations, just because they are now two different populations. All right, next one, bottleneck effect. So we have a population size, and then it decreases a lot. What happens is, in the end, we can have no variation left in that population. So what will happen when we had that original population, it was very big. The new population is very tiny. Based on those two things, one of the characteristics of that large population died off. What happened to it, I don't know. All right, but the ending population, there's no variation left. Okay, so what will happen over time with a bottleneck? The population probably will go extinct over time since there's no variation left. No variation means no natural selection. No natural selection means no evolution is going to occur. So here's an example, and you guys can see it. They literally use a bottleneck for this. We have different traits in here, and we empty them out, and you guys can see the surviving ones. We got a little bit of white, and we got a good bit of blue, but all the yellow ones are gone. The Florida panther is an example of this. It had lost its habitat because of roads being built in Florida. A lot of them became roadkill, but yeah, kind of sad. They hunted them before the 1950s, so that killed a lot of them. And hunting, obviously you want the biggest panther, or if we're talking about deer, you want the deer with the largest rack. So a lot of those good genes actually die out because of that. All right, so we're good here. Oops. I want to go over this picture with you guys for a second too. So just continuing on with the Florida panther here. In 2008, obviously, because of the bottleneck, they were an endangered species, and there was only 6 remaining. So now, in 2016, we actually have 230. That was the most recent data. So how did this number go up? If we only had 6 remaining, I mean, my goodness, it's almost done there. It's going to go extinct. What they did was they hybridized the Florida panther in order to increase the population. They tried to find a species that was really close with DNA, was really close with chromosomes, and they found the Texas cougar. So the Florida panthers, if you guys see one now, which you probably won't, since there's just over 200, they hybridized them just to try to save the species. So it is a hybrid animal now. All right. Okay. So one more example of genetic drift, guys. We have a prairie habitat. We're going to talk about some birds in that prairie habitat. Now, they looked at the prairie chickens in different parts of the United States. They looked at them in Illinois and Kansas and Nebraska. So there was a loss of habitat in the Illinois one. With the ones that survived, the prairie chickens that survived, they found that their eggs were not hatching. Only about 50% did because of this bottleneck. So in Illinois here, if you guys can see, between 1930 and 1960, we had about 93% of the eggs hatching. But in 93, after the bottleneck, less than 50%. But where there was no bottleneck in Kansas and Nebraska, the eggs were still good. They were still hatching at a very big rate there, okay? So that is what happens when we have a bottleneck. Obviously, we did not know that these eggs, weren't going to be hatching, but the traits that were left over after the bottleneck were traits that were probably not as good for egg hatching as before the bottleneck. Okay. All right. So we're good here. We went over that one already. All right. Just a overall summary of genetic drift, and then we'll stop here for today. Okay. So first one, because of the alleles, if it's a small population, genetic drift is going to have a major effect on that because we're losing some of those alleles, whether they're dominant or recessive. It's going to change the numbers a good bit. Genetic drift can cause our alleles to change, or the prevalence of those alleles, All right, they will change in the population. We can lose genetic variation. You guys saw that with the bottleneck there. And then lastly, the genetic drift, if we do have that bottleneck, maybe the harmful alleles, and I know the prairie chicken, it wasn't really harmful, but less than 50% hatching, that could lead to the end of a species or majorly reduced population numbers in that species over time. Okay, but overall, genetic drift still does lead to natural selection, but it changes natural selection just because of the genes are randomly being eliminated. Okay, guys, so next time we'll start with gene flow, and I think that's enough for today. See you next time.